So in this, I'm going to give a little bit of historical background, a very, very brief overview, because I'm only supposed to spend 45 minutes, of some approaches that are not going to be the subject of this class, but are the sort of interface between what's currently going on in quantitative genetics and how it relates to this evolutionary quantitative genetics. And then I'll illustrate one of those in just some of my work from my own lab. And so I think what many of us are interested in is understanding this diversity of life, and this has now become a joke that everybody begins their talks this way. But I think this is real. This is the central problem of biology, is to understand both biological diversity in terms of the number of different kinds of organisms there are, but also because asking questions about how they look. There's a good guy for and so understanding why organisms are the way that they are. But at some level, we think it must be related to this as well. We also need to understand why the molecular structures that make up organisms are the way that they are. And so evolutionary genetics has sort of taken these two separate paths, and that's going to be one of my themes for today's lecture, is that there's still an ongoing path of trying to understand sort of the molecular level, and then this course is mostly concentrating on the phenotypic level. They're both based on genetic ideas and the same kind of evolutionary models, but it's the linkage between them that is still the future here. And it's natural, I think, in science, is that different parts of science have different emphasis areas that are working. And so without going into this in any great detail, we have different sort of levels of organization, from genes to the diversity among organisms. And Mendelian genetics, when it came out, is basically trying to understand the relation, well, basically what genes are doing in terms of transmission just by looking at phenotypes. And then molecular biology is focused on the translation between genes that make phenotypes. Population genetics ignores phenotypes, but just essentially has an idea of what fitness is, and then how it changes genes. And then quantitative genetics really focuses on this loop up here, especially the relationship between the phenotype and fitness or ecological context, and that's going to be a lot of what you work on in this course. And I think the unique thing about this course is then trying to tie that to these ideas of diversification. That is, how do these microevolutionary processes go to the macroevolutionary? So that's great. But for the most part, for most of its history, quantitative genetics is assume that there are these things called genes, but that they have aggregate statistical properties, and we don't need to worry about the details. And that may still be valid, but and then we have comparative phylogenetic methods, which is really interested in making these connections among phenotypes and diversity at one level, and then sometimes you use genes to infer phylogenies, but not a connection. So one of the things I want to emphasize is that, as far as I know, evolutionary quantitative genetics is the only coherent conceptual framework for all of biology that puts all these things together. But so we have a single conceptual framework for going all the way from gene to total biological diversity. But what we don't have still, and this is where you are all going to make your contributions, we don't have all these linkages are not completely solid. Okay? So this is the right way to be approaching the questions, but we don't know the details. So part of this is, again, historical. It goes back to the very beginning of genetics, in which basically everybody who started working on biological questions around the turn of the century was interested in evolution and diversity, but there became a schism, basically, based on our what kind of knowledge we could have. And so genetics divided into two parts. One, people who studied what we call Mendelian genetics, which was to understand what is the material basis of inheritance, and the other was to study why organisms look the way that they do, and that became statistical genetics, because that group appreciated that we weren't going to be able to figure out the underlying genetics, but we could still understand genetics by looking at the resemblance between relatives. And this schism is continued. Statistical genetics went on to form the basis of agricultural genetics and then population genetics. And then Mendelian genetics went on to form the basis of molecular genetics, because, again, the focus on the material basis of inheritance. And these fields now are starting to sort of re-merge occasionally. And so one of the first re-mergings was what's called Evo-Devo especially in the 1980s, where people interested in the evolution of developmental processes and were able to combine sort of our emerging knowledge of the functional basis of development and to create a synthetic area there. In the meantime, quantitative genetics tried to start becoming a little bit more functional and molecular through QTL mapping that I'll talk about in a second. And then these sort of mergings are going on. Population genetics has continued on this molecular evolution thread into modern genomic analyses. And then QTL mapping hasn't worked out as people hoped, and so has now progressed on to GWAS, which is then, which we'll talk about, this genome-wide association studies, which now has elements of other genomic components. And then finally, we're at this major crisscross where all these fields are merging together because molecular biologists understand that they need to know something about population variability in order to address the questions that even within human disease, for instance. And evolutionary biologists recognize that in order to answer their questions, they need to know more about the genetic underpinnings of how things work. So I just wanted to emphasize what I think is this fundamental distinction between this schism that occurred in genetics that I think is really important for your thinking as you progress on through this week and hopefully through the rest of your careers. And it comes down to what the fundamental genetic unit is and how you think about genetic information. And so in this tradition of Mendelian and molecular biology, the fundamental genetic unit is the locus, that is the gene. And that comes from a more or less typological way of thinking that there's a wild type, or now we have the reference genome, and that other things are mutant from that state, okay? And so because of that, you can say there is a gene for X or whatever. In quantitative genetics, though, the fundamental unit is really the allele. So what actually exists in an organism is some DNA sequence, okay? And that is what creates function as an emergent property of that sequence. And different alleles have different DNA sequences. And part of the reason why this is important is because there can be variability in effects at the level of alleles, and you don't really need to think about a gene having a function because multiple alleles at a single gene can have divergent functions and variability in their functions, okay? And that turned out to be very important, especially for multivariate quantitative genetics. So one example, I think this is the most important example, is actually in pleiotropy. So in molecular biology, pleiotropy, as you know, means that you have a single gene that influences more than one trait. And so in molecular biology, you would tend to measure this by having a mutant effect that affects more than one trait. And so you would say that pleiotropy is a property of a gene or a locus. But in quantitative genetics, pleiotropy is a function of allele, and pleiotropy can be variable from alleles to alleles. So you can have an allele that affects more than one trait, or an allele at the same locus that doesn't, or affects a larger combination. That is, there's variability in pleiotropic effects, and it's that variability that will lead to the evolution, for instance, that Adam will talk about in terms of the evolution of the G matrix, for instance. Another example that we don't have time to go into is in epistasis. And again, in epistasis, in molecular biology, you tend to think of epistasis as a mutation that when that mutation exists in one gene, it blocks the appearance of mutants or the phenotypic effects of mutants at another gene. And it's epistasis literally means to stand upon the other effects. And so an epistatic locus stands upon the effects of the other loci. In quantitative genetics, though, we have a much more broad definition of gene interactions that can involve any kind of possible nonlinear interaction between the genes. And so what's happening now as these fields become more and more similar, people use the same words to describe what turn out to be fundamentally different phenomena. And so it's always important to appreciate what you're talking about. I don't have much time for this. I just gave a talk at the International Quantitative Genetics meeting, And I started to make a cartoon history of a version of the history of quantitative genetics, but I thought it'd be fun just to show you a couple slides from that. So back in the day, so you recognize this guy. This guy is Russ Landy. So these guys started taking us on a wild ride through quantitative genetics and in the early 70s. And a lot of what you'll be doing this week is based on venturing through Landy land. And so we started making progress, and you'll spend a lot of time working with this set of equations, the delta z-bar equations. And really this set of work by Steve and Russ helped create the fundamental conceptual foundation for what we're doing. And so we reached sort of the pinnacle in 1983. They're very proud of themselves with this delta z-bar equation in this form. And you'll be doing work figuring out what G is and what beta are and how these things combine. There became critics of some of this work and the underlying assumptions, Michael Torelli being chief among them, that caused problems. And then people started trying to figure out the molecular basis of quantitative genetics, and that leads to more problems. So Bruce Walsh introduced this idea of a wombat, which is a waste of waste of money, brains, and time is a project. And so a lot of molecular quantitative genetics that I'm going to talk about for the rest of the class is essentially been a wombat because we haven't learned very much. So this is Bruce Walsh, and he was trying to save the wombat, but that's the end. Okay, that's as far as I could make it for the cartoon history. So again, we have this equation that other people will be talking a lot about, but the idea is this G matrix thing somehow encapsulates the genetic associations among traits and the relationship between them, and it's based on the idea that traits that we observe here at the phenotypic level are going to be underlined by some complex set of genes, and individual genes can influence more than one trait, that is the pleiotropy, or there could be tight linkage that leads to associations between traits. But somehow there's some translation in between, and that's what we still don't know a lot about. So getting back to this idea of that the allele is important, we frequently think about sort of a single locus, one locus or another locus, then having an influence on a wide set of traits that we usually describe by Z in the Chicago School of Quantitative Genetics. But in reality, what we have is a set of alleles at loci that each of which can influence many different traits, and so that there's a combination of allelic effects. So in practice, what this ends up looking like is that you can have alleles at one locus and alleles at another locus, and they can have a distribution of pleiotropic effects. This is alleles. I misspoke. This is allelic effects at one locus on trait 1 and allelic effects on the same locus on trait 2. So you can have a distribution of possible pleiotropic effects, and that those things can then be molded by selection. And Adam will probably talk a lot more about this in a second, this afternoon, or tomorrow afternoon, I guess. And so these are influenced by mutation properties. And so whatever the final thing that we're looking at is some balance between mutation and selection. And so what I'm going to just focus on for the end here is what are these little dots? And what does it mean to have variation in pleiotropic effects? And how are we going to discover what these things are? So we somehow want to find the alleles that matter here. And so why do we want to do that? Well, I think a lot of people want to do this because they're just interested in biology. That is, they want to understand how organisms work. So you want to find the functional biology of adaptation. If you see mice adapting to high altitudes, you'd like to know how their hemoglobin is changing, just because that's an interesting question in biology. But once you find genes that matter, then you can actually use the approaches that for instance, Joe works on and has developed to use molecular population genetics to understand the history, the evolutionary history of these specific genes and alleles. And that should give more insight into, for instance, whether or not there's been integration between species. Does adaptation usually occur through hybridization and migration, or is it through de novo mutations that arise independently in populations? And I think we are learning a lot about that by using genomic information. More fundamentally, and this has not been the focus because it turns out to be a really hard question to do, we need to actually understand what the distribution of these effects are in order to use these evolutionary quantitative genetic models effectively. And we know almost nothing about this, okay? And it's because we still aren't able to do this work very well. And then eventually, if we figure this out enough, then we can actually ask questions about how the whole genetic system itself evolves. That is, how is the functional biology of organisms structured by evolutionary processes? Why does the molecular biology of organisms exist the way that it does? And we still, again, are not equipped to address these questions at all. So I was tasked with basically introducing all of this molecular approaches to quantitative genetics in 5 minutes. So that's we're going to do the best we can. All I'm going to do is give you just a very brief overview of the approaches that are used. There are a whole other week-long courses that you can take in order to learn this material. And during lunch and dinner and things like that, I'd be happy to talk to you more about what these different things are. But suffice it to say that we're trying to figure out the answers to these questions. And also suffice it to say we're doing a really horrible job of doing that, I would say. So I'm going to emphasize 3 different approaches. The one that developed most directly, really starting in the 70s, but had its heyday, people are of course still doing this, in the 90s and the turn of the century, is quantitative trait locus mapping, or QTL mapping. And the idea of QTL mapping is you use a controlled cross between known strains and then basically use segregation analysis of markers to try to identify the genes that influence a trait. This has a lot of strengths because you're controlling the genetic backgrounds, and so the genetic inferences that you're able to make are more powerful than in many other systems. And because it's a segregating cross, usually there's strong linkage between the markers and the putative QTLs, and so you're more likely to identify something as having a genetic effect. But that's also a weakness, as we'll talk about in a second, is that when you don't have as many recombination events, and so the likelihood that you'll be able to narrow in to a specific gene is quite low. And because of that, if you have large blocks that are linked together, there could be many genes that are important all in that block. And it really goes down to what the actual genetic architecture is. Are there thousands of genes that affect a trait, or only a few? And so most of the, when this work started, and this is actually a general feature of molecular quantitative genetics, most people expected or hoped that there'd be a few genes that are influencing quantitative traits. And a lot of the methodology is built under that assumption. And when that assumption is false, which it appears to be, then these methods are not particularly useful. Another approach that we'll briefly touch on is called genome-wide association mapping, or GWAS. So here, instead of having a controlled cross, you use naturally segregating variation, and you analyze the markers of many different individuals in a population and just look for an association between the markers that you observe segregating in a population and the trait that you're interested in, and then make an inference about if there's a positive association between those things, that there must be a genetic basis to the trait. Its major advantage is because you're not just limiting it to a couple of genotypic backgrounds, you could potentially find all the relevant alleles. And the most useful part of it is because you're relying on population samples, you have this long history of recombination that's going to break up associations between different markers, and so that will give you much higher precision within the genome. The weakness is complexity. You're analyzing a whole population with all kinds of genetic effects, and so nothing's controlled, including the environmental variation and all sorts of things, depending on the population you're looking at. And so that can be quite complex. In, there's Joe. In other models, you can, say in model systems, you can control some of this background and make repeated experiments. And for natural populations, you have to know something about population structure. Otherwise, your inference, anything that's different between the populations, if you don't recognize it, is going to be associated with traits that also happen to be different between the populations. So you have to account for that.
Hello, am I back?
You're back. We can hear you, Joe. I think
Can you see me?
we can see you. And I think the plan is that I'll finish my lecture and then we'll flip it over to you, if that'll work.
I don't think there's time for me to give a lecture. It was problems with my computer at this end. I switched to my laptop, and maybe we'll get better connection.
Well,
I have been hearing your lecture, which is a very good lecture on a different topic.
yeah. So there would be time for you to give your lecture if you can do it, because I'm giving the lecture I was going to give later. So there'll be time in the schedule.
Oh, okay. Go ahead then.
Okay, good. I'm giving the talk that I was going to give at 11 o'clock our time. So you can give it after me.
I won't interrupt it then.
Okay. All right, so the final method that I'll talk about is a different approach that I think is unique under evolutionary approaches, which is what are now called evolve and reseek things, where we actually do selection experiments and use the power of the way that we understand selection to make inferences about genetic changes. And the strength of this is that you usually have some ancestor, so you have a null hypothesis at which to compare to see what's changed. And also, it should be more limited to the genes that you think are interesting from an evolutionary point of view, as the genes that actually respond to selection. The problem is that you'll only be looking at genes that are under selection under your specific circumstances, not all possible sources of variation. And so that's potentially limiting. And also, as we'll see in a second, the genome is large, and genetic drift is really a nasty thing to try to distinguish against. So the primary challenges here are that, as I intimated, the genetic architecture, that is the actual number of genes that are influencing the trait, completely determine whether or not your method is going to work. And of course, we don't know what that is until we start doing the experiment. And so there's a bit of a circularity in designing the appropriate experiment for the question, because you don't know what the answer is until you start doing it. And it's, I think there's a long history of people over-interpreting their results because they do a cross, like a QTL cross, that will, based on one set of assumptions, and then and then make inferences on that basis. I'm sort of loath to mute that again, but that's the way it goes. And then there's this trade-off on linkage and how it influences whether or not your inferences could be made. And so, in particular, if there's high degrees of linkage disequilibrium, then all your markers are going to be in strong association with the underlying quantitative trait loci that you're interested in. So you're very likely to detect a genetic effect, but you're very unlikely to be able to specify exactly what is causing that effect. Whereas if you have very low linkage disequilibrium, for instance, in a GWAS thing, then you have very low power to detect anything, because you don't have coordinated sets of markers, but you have very high precision. And so this is sort of the QTL world is over here, where things are in high linkage disequilibrium. You're likely to be able to detect genes, but you won't be able to say where they are. And GWAS is down here, where you have very low power to detect things, but if you do detect something, it's likely to be quite narrow in the genome. And so this really goes to the question of how big the samples need to be in order to make these things work. So this is from this paper on missing heritability that talks about another element, which is what the allele frequency is that's segregating in the population. So effects that are at high frequency, especially in GWAS studies, are going to be easier to detect, and things that are very low frequency are going to be very hard to detect. So things that are at low frequency and have small effects are essentially impossible to detect in quantitative genetics. In human disease, we're not detecting things, as we'll talk about in a second. So now the thought is that it's probably a lot of human variation is generated by rare things with large effect, which is the hope because they could still be detected. There's some possibility, especially within natural populations, that this is the world that quantitative genetics lives in with rare variants of small effect. And if that's true, then actually identifying the molecular basis of quantitative traits is going to be very difficult. Okay, so I'm going to introduce these approaches again in a very cursory fashion. But QTL mapping, actually all mapping works on exactly the same principle. It's very straightforward. Doesn't take a lot of thinking to do it. But then, of course, in practice, there's all the statistics and bioinformatics that's built around there that makes it seem more obscure. But in general, what happens is that you have a set of lines that are somehow genetically variable. In this case, these are recombinant inbred lines that I'm showing on the slide. And you can imagine just scanning across each section of the genome. So let's take this first section up here. We just scan across on a marker, and there's either going to be orange or blue at that given marker. And you just divide the samples, the different lines, into two groups. You're either in the orange group or the blue group. Okay, and you measure all the phenotypes of the blue guys, all the phenotypes of the orange guys, and you ask if they're different. If they're more different, the more different they are, the more likely it is that a gene associated with that marker is actually causing that difference. And that's the essential nature of the inference. You go down to the next marker down, and then you have a different group of red and blue because of recombination has moved markers around, and you make that same inference. So if there's essentially no difference between the red and the blue, so here's the blue and the red and orange distributions are overlapping here, then there'll be a very low likelihood that there's a gene that's causal there, where here, when we look at the distribution of the different lines for orange and blue, we see that there's a difference between them that we can detect, and so we can do something as simple-minded as a t-test between these and then use that as a statistical measure about whether or not they're different. So imagine doing this at every marker that we have in the genome, and the first studies that did this for a few dozen markers, and now we can do it for tens of thousands of markers, and then you just scan across the genome and basically do 10,000 t-tests. It's more sophisticated than that. Actually, it's gotten less sophisticated over the years. The initial QTL stuff that became sophisticated was you had fewer markers, and so you actually used the linkage map information to make an inference between the markers. We've gotten away from that as markers have become cheaper to generate. So basically, you end up getting these plots where you have some significance threshold, and then you say there's a couple of important QTLs there. So this could be any trait. This paper from Matt Rockman in Nature is actually talking about RNA transcripts as a trait. So you can imagine if you measure transcription of every gene in the genome, there's tens of thousands of traits that you're looking at. And so if you collect those, so if you do this on one trait and then another trait and then another trait, over time what you could do is if we have all these gene locations down here, we can then say, we can build up a little map of every, in this case because it's a transcript, it's related to a gene that's in the genome. So we can say, where is that gene expressed? And then where does it map to in terms of an important QTL? And this is essentially a pleiotropic map of gene transcription, okay? So what this means is that this G site, all these different transcripts are influenced by allelic variation at the G locus, okay? So there's a pleiotropic effect. So this, for instance, might be a transcription factor that is having an influence on many different genes. So this is one way of thinking about pleiotropy in this QTL context, is how many traits map to the same location in the genome. So how many, if we do trait after trait, how many of them map to QTLB? So it's a way of trying to pull apart the pleiotropic relationships that you'll be talking a lot about through the genetic covariance, and trying to get them aligned to a single location in the genome. So that's the basic idea. It essentially just takes genetic mapping and puts measurements on it, and then scans through the genome. So as I said, QTL analysis has been used a lot. There's actually very few examples in which it's been particularly useful at cloning the genes underneath there. There are some examples, but not great. And that's because if there's too many QTL, the effects are small. And then these regions that potentially could have the marker in them, even though on the scale of the whole genome they're fairly narrow, there's usually several hundred genes in a given region. And so it takes a lot of work to follow up. You have to be very motivated to figure it out. And then you have to do the functional analysis. So it has been done, but not as frequently. So even in agricultural systems where people are usually highly motivated to try and use these approaches, it hasn't been done that often. So if the trait is simple, only influenced by a couple of genes with very large effects, then you can do it. If that's not the case, then it usually fails. So people said, okay, that's not working, let's invent something new. So this genome-wide approach started appearing in the late 90s. And the idea here is that rather than working in a controlled cross, you can use a segregating population. And this is especially applicable to human populations because it turns out doing controlled crosses is frowned upon. And so if we want to identify the genetic basis of, say, a disease like multiple sclerosis, for instance, it's a very hard thing to do in a human population, but we can identify many patients that have a given disease, and then we can identify controls that don't have it. And then if we look at genetic markers within those two groups, again, it's the same idea that we were doing before. We have disease, non-disease individuals, and then we just scan through every location in the genome and ask whether or not that particular thing distinguishes them. Okay, so instead of thinking about disease and non-disease, what you end up doing is taking a given site of the genome. So at this site, you have some individuals that have an A nucleotide and others that have a T. So those now are your new groups, A group and T group, and you put everybody, you either have A or T, and then you measure the frequency of having a disease if you have the A or T. So if the frequencies are the same, then you would say that it's unlikely that that nucleotide influences it. But the more the proportions of disease states differ between those two groups, then the more likely it is that that nucleotide has some influence. And so that's what you end up measuring, okay? So this is an outcome, and there's many, 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 many of these studies now that are coming out, but this is a typical kind of results. I guess now it's old, 3 years ago. This is now a small study, so this is of a human population, and this is the whole human genome surrounded. And so what you end up doing is each one of these things is a measurement of the of basically this likelihood, or really it's the p-value, the negative log of the p-value. And so everywhere that there's these red lines, that's a site in the genome that is having some influence on the probability of having a disease. So in this study, there's about 15,000 subjects and 24,000 controls. They looked at a small number of markers, 160,000 SNPs, which is now small. And they identified 103 loci. And this is really important. Even though they had what for an evolutionary geneticist would be a huge study and identified a lot of loci, they're only still explaining 28% of the variation. So this is the theme that's emerging from all of human quantitative genetics, is that as the studies are getting larger and larger, hundreds of thousands of individuals start doing whole genome sequencing now. And what we find is that there are many, many hundreds, if not thousands, of sites in the genome that are influencing most quantitative traits, and they still explain a very small fraction of the genetic variation. Okay, so complex traits are actually complex. And so this raises the question for this course is whether or not we need to know any of this stuff in order to apply just normal quantitative genetic models that are built on this assumption anyways. Yeah? These are all first order, yes, no interactions. You mean like epistatic effects? Yeah.
So I just asked him, are these all first order effects in this study?
Yeah, if that's what you mean by first order, it's just the linear additive effects. And that's usually what's looked at. You can imagine how many interactions there are among 160,000 SNPs that you'd have to look at. So this is the classic trait of quantitative genetics. It's the first quantitative trait analyzed by Francis Galton in 1889. It's human height. This is the first regression plot. Regression, as you may know, was invented to solve quantitative genetics. Matter of fact, almost all of modern statistics was invented to solve problems in quantitative genetics. R.A. Fisher invented the analysis of variance to do it. And so it turns out that studying a few tens of thousands of individuals for human height is not enough. Let's just concentrate on this one. So this is this prediction which is holding true right now. And they're estimating, based on their initial study of how many genes they could detect that were influencing height, how many individuals they would need to study. And this is the cumulative variance explained. So in 2010, they predicted that if they studied 730,000 individuals, that they could get up to explaining about 4% of the variation in height, okay, for 270 loci. And so they've since redone this analysis, and I can't remember, they're up to the prediction of about 800 loci, but it's looking like it's going to be millions of individuals studied and thousands of loci influencing human height, for instance. So sort of by necessity, if you have 1,000 loci influencing it, the average effect is going to be fairly large. I mean, fairly small. But they don't appear to be many loci that have large effects. Now, we do know that there are loci with large effects on human height, for instance, loci that lead to dwarfism. But those are rare. And so this goes to the question of where does variation emerge from? Is there some fairly rare loci of large effect that contribute a lot of the variation? And if that's true, then sort of our normal assumptions about the distribution of effects of loci, for instance, being normal, might not be very good. But from an evolutionary standpoint, we actually don't know still what contributes to the response to evolution. Is it all these small effects that are quite common that are pushing the whole distribution forward, or is it occasional rare variants that are then swept? And to some extent, people who are interested in macroevolutionary patterns tend to have one viewpoint that these rare variants of large effect can be more important, and microevolutionary biologists, especially evolutionary quantitative geneticists, tend to concentrate on the common segregating variation. I tend to fall into that camp as well. So I won't dwell on this, but this is now turning out to be a major problem. The idea when all of this started is we do this mapping, we'd find some nucleotides, it would point the way to a gene, and then we would figure out what's going on, and then for human disease studies, then you'd invent some drug that would help. That's clearly not going to be the case. These effects are very small, they're weird, spread throughout the genome, and so it's a very difficult course of study to, you can do the mapping, but then to actually figure out what the genes are, there's a lot of bioinformatics. People are now combining different sources of functional genomics. You have expression data, you have epigenomics that you need to be concerned about now. These methylation effects could be important for causing the effects of the allele. And so all of these things need to be combined. So again, in an evolutionary genetic framework, we're frequently not prepared at the level of effort that's necessary to make all this work. Okay, I'm going to skip through this part. Just talking a little bit about systems biology here. So essentially this quote from Lewontin is one of my favorite ones. You can always, Lewontin's sort of like the Bible. If you want to have some point of view, you'll find some quote from him that will support you, especially if you want to be a curmudgeon. There's going to be some negative Lewontin comment somewhere. But just a lot of wisdom there. So basically the idea is that, well, we want to figure out what how to understand the genetic basis of adaptation. And he says the assessment of adaptive variation will depend on understanding the relationship between the gene and the organism that far transcends any present knowledge of development, physiology, and behavior. That is, if we actually want to do what I've just been talking about, we have to, it demands that we answer every other question that now lies open in biology. So to really do this properly, it is the question of biology, is what is the relationship between genotype and phenotype. So it's difficult. I'm just going to finish up here very quickly, just showing you an example of this evolve and reseek approach using our own work. So this is the kind of thing that one could start trying to do if you had some functional biology. So one of the things that we study is stress response. And so here's a stress signaling pathway that's mediated by the insulin-like signaling pathway. I study worms, nematodes. And so we know a lot about the molecular biology of this that's been figured out over the last 25 years. Basically, you have a receptor, you have a signaling cascade that goes down to a transcription factor that turns on stress response genes. This is sort of a canonical way of thinking about this. But this exists within a larger network of stress responses that all have their different sort of inputs and signals, and then are integrated together through a broad set of transcription factors that all interact in some complex way. And so what we would expect is that there could be strong pleiotropy through this kind of thing. So for instance, if you're interested in starvation being mediated through this insulin-like process, and you're also interested in oxidative stress, that they all ultimately end up impinging on a similar set of transcription factors in the middle. So this is the functional biology of pleiotropy that we might anticipate. So we've been working on this for a while using nematodes as a model system. So this is a species, Caenorhabditis romanii, that's closely related to C. elegans. It's a better system for doing quantitative genetics because there's a lot of natural genetic variation. And they're also an outcrossing species, males and females, so that they have sort of more normal population genetics. So we've been selecting them in a variety of stressful environments. I'll just talk to you. This is Kristen Sinkic and Rose Reynolds, a graduate student and postdoc that were in my lab that did a lot of this work. And so we've been selecting them in these different stress environments. I'll just talk about this where we selected them under acute periodic oxidative stress and heat stress. And so this is only about 10 generations of selection, 30 generations total in the lab, and it's only replicated a few times, but very large population sizes for an animal experiment. And so what we find, this is everything relative to the ancestor. So one advantage of worms is that we can freeze them, so we have a fossil record that we can bring out of the freezer and compare at the same time. So this is the response relative to the ancestor. And what we find is that in the controls where we're not selecting them at all, they don't change over time, so that's good. And then we select them either to oxidative stress or to heat stress and then test them across both environments. So this is a multivariate trait. So when we select them for increased heat stress resistance, they increase their resistance, so that's good, selection works. They don't have a correlated response to oxidative stress. And when we select them to oxidative stress resistance, they increase in oxidative stress, but those guys only have a very minimal increase, non-significant increase in heat stress resistance. So we expect there to be pleiotropy based on the system, but we don't observe a lot. So I'll skip over this part. So we are using genomics to try to identify what's going on. So this is all the changes from the ancestor and the control populations. So everywhere there's a dot, that's a significant part of the genome that's responded. So we see that there are parts of the genome that seem to be involved in adapting to the lab. And then these outer rings are the heat, in red, and in blue are the oxidative stress resistance guys. And so what we see is that basically the whole genome lights up somewhere. So when we look at a fairly stringent criterion of genes that evolve in one environment, but across both replicates, so we have to have replicated evolution within a gene and then not showing up as changing in the control, then what we see is that there's about 177 genes that are significantly respond to selection, and this is very conservative actually, in the heat stress, and about 134 that evolve in the oxidative stress, and there's very little overlap between them. So this is one way of using this approach to actually test this hypothesis of pleiotropy, is to do the selection and then see whether or not there's correlated responses to selection. So we don't see that. And we can also look at this at the level of gene expression, where we see many genes are upregulated in the oxidative stress, and a smaller number, but still quite a few, significantly upregulated in heat stress, and a little bit of overlap between them. This is the control. So by and large, what we find is that when we select on these traits, we're able to separate them. So overall, for this result that I just whipped through, we see that even though we see these shared functional pathways in terms of the evolutionary response to selection, that's not what we're seeing. We expect there to be this highly coordinated pleiotropic set of things, but the response that we see are in these non-pleiotropic alleles, getting back to this theme of the beginning of this talk. I didn't have time to talk about it, but we'll also look at the evolution of phenotypic plasticity. So overall, we can start using this kind of approach to break out the quantitative genetics, in this case, this model of stress response, which is this highly correlated set of phenotypes, and try to identify the genetic basis. But what we find is the same thing that everybody else is finding, is that there's lots of genes involved. It seems to be highly complex. Figuring out exactly what matters is not trivial. And then, again, as a basis for the rest of this course, does it matter what this is? Or is it perfectly fine to just say we have these concepts of additive genetic variance, additive genetic covariance, we can understand how selection works. There are things that are going to be changing at the genetic level, but we don't need to worry about that unless we're really interested in the functional biology of that. And I'll stop there.